The next of the three processes of the central dogma of molecular biology is transcription. After replication, we have produced copies of the DNA. It is important that we produce RNA in order to produce the proteins later on, alright, that we need. Transcription can also be given the name DNA directed RNA synthesis, because its source again is DNA, but the product that we get after transcription is RNA, not anymore DNA, which is what we saw in replication. I would also like to mention that the RNA that we produce here in particular is mRNA, or messenger RNA. There are several RNAs that do several different functions, but the mRNA is the one that is later on read by the ribosomes to produce the amino acids and later on the proteins. So we will get to the different RNAs later, those which are important. And transcription is also composed of an initiation, elongation, and a termination phase. But after you produce the mRNA, once termination has occurred, there's also post-transcriptional processes that which have to be done prior to translation. One thing that we have to note is there is a certain term as fidelity, and fidelity here means fidelity to whatever we need to produce. And there is a certain weight on the fidelity of transcription, alright, which is not that heavy, alright. For transcription, it's not that significant, but for replication, it is very important. What do I mean? It means that if you produce one wrong DNA, then it would produce a lot of mayhem in the particular organism we are talking about, instead, as compared as to when you just produce one wrong RNA. If we go back to the products of replication, which is DNA, if you have this one is one correct DNA, then you're going to make 1,000 copies of correct RNAs, alright? And if, for example, you have, out of those 1,000 RNAs, you have like 10 wrong RNAs, but you still have like 990 correct ones. So what process did you do here? You just transcribe this correct DNA, alright? But if you replicate a wrong DNA, you're going to make thousands of wrong RNAs, and automatically, if you have just one DNA here, but it's one wrong DNA, and you're going to transcribe it, going to use it as a template for 1,000 of wrong mRNAs, you're going to cause a lot of damage. So there's a lot of weight in the fidelity when it comes to replication as compared to fidelity when it comes to transcription only. Alright, so now we go to the initiation part of transcription, but I'm going to introduce again a DNA template here. So here, we're not going to name these as lagging or leading strands. Okay, that's for replication, but for transcription, we're going to name the two strands as templates or antisense and sense. The sense strand doesn't make sense at all because it's not going to be used in transcription. The one that we're going to use is the template strand. Alright, that's why it's named as template. And the direction of transcription is again from the 5' to the 3' direction, just like of the template strand, just like in replication, but that was for the leading strand. Now we call this as the template strand. So for initiation, there's just one enzyme. It has a generic name, right? It's simply named as a holoenzyme, and it's composed of two parts. It has the core unit, which participates in elongation, and the sigma, which participates in the initiation. Alright, so supposed to be the DNA strand is closed again. So obviously what happens in initiation is you open up the strand. So the sigma strand would open up the strand on a particular area. That particular area for initiation, which the sigma unit opens, is known as the promoter area. And the promoter area would signify or would signal the origin of transcription. So for example, I have the sigma unit here. It starts here. So that's the part that it would open up, and that ends initiation. Alright, just simple as that. Now, we have the core subunit here for the hollow enzyme, and that would take part in the elongation part. Alright, so just like in replication of the leading strand, it would slowly introduce one by one the nucleotides. Alright, and that's simple as that. Okay, that's simple as that. So when you reach a particular area of termination, then there's another enzyme that would participate, usually, but not all the time. What do I mean? So termination in transcription can happen in two ways. First is that there's an enzyme, alright? This one, the enzyme here is known as the raw enzyme, alright? Or it could be drawn simply using this one, the raw. So if the termination is raw enzyme dependent, then of course it would use that enzyme. So the raw enzyme would detect that the site of termination, and it would finally end the mRNA there. Just that simple. But here, if we're not going to use the raw enzyme, which is the second variation of termination, it would be, well, first it would be raw independent, obviously. And what would signal its end? Well, here in the antisense strand here, there's a particular formation of a hairpin loop, and the one that made this one responsible is a lot of CG and GCs here. Remember that cytosine and guanine, they form 3 hydrogen bonds as a base pair, and if you have a lot of those colliding, joined together, or a lot of them nearby, it will now form a hairpin loop. That's a lot of interaction. That's a very strong interaction, and it would close out like that. Now, the core enzyme, or the hollow enzyme, cannot interfere with this hairpin loop. It cannot go inside that, and thus it would end at the part before the hairpin loop, and termination would now take place even without the raw enzyme. And so with that, you now finally have mRNA. However, as I said, it's not yet the end really of transcription, because there are also post-transcriptional processes that take place. Well, first things first, the mRNA that you produce here have this particular adjective, which is premature or precursor. I'm going to write it because it's not that clear. Precursor or premature. So meaning, it's not yet the mRNA that's going to really take place in translation. So what has to be done there in order for it to become the mature RNA that we have here? Alright? There's a particular name known as splicing. Alright? Splicing. So what happens is that in splicing, you remove certain parts of the mRNA which are not really going to be read by the ribosomes later on. So those portions that are not really important are the introns. Alright? They are just inside, they're not really there to be read later on. So splicing removes the introns, alright? It removes the introns, then it joins these exons together. So all the exons will now be part of that mature RNA that we have, alright? In addition to splicing, we have two additional processes. First would be taking place here, and the second would be taking place here. So the one here at the 5' end, which is the starting point, is what we call capping. And well, since this is the starting point, just like a head of a human person, you put a cap there. Alright? So you put a particular cap there. Alright? This is the cap. And the composition of the cap is a particular nucleoside. Alright? And it is a methylated one. And here at the 3', we have polyadenylation, meaning you have many adenosine nucleotides. Alright? So this is like A, then a lot of that. So you have a long poly A tail, alright, at the 3' end. So the purpose of both of this is in order to protect the mature RNA. Because remember, the moment you get the RNA already, what is it going to do? It's going to go out of the nucleus and travel its way through the RNA. But one thing we have to note is that in the cytoplasm, alright, I'm just going to draw here. So we have now the RNA here. So the RNA goes out to the cytoplasm in order to meet, for example, the ribosome here. But when it goes out, the cytosol or the cytoplasm contains a lot of nucleases, and these nucleases would target the mRNA and would try to break it down. So what the cap would do, and also the polyadenylyl tail, is it's going to protect this RNA. So what's going to do is that the nucleases would chomp down on these ones, alright, so that it would then reach this one. So the tail here will serve as a sacrifice in order to prevent this actual part from being degraded. And this tail here, this methylated part, would sort of be immune to the nucleases, so that this part will not be degraded by the nucleases. So basically, the cap and the tail would serve as protection to the RNA against the nucleases in the cytosol. So that's it for the transcription part, and the product again is a mature mRNA.